Hi, this is Dr. Kat Vlies from Central New Mexico Community College again to discuss the digestive system, this time with a focus on the small intestine. And this is video H in a series of many more to come. The most proximal portion of the small intestine that connects directly with the stomach we could refer to as the duodenum, or you could say duodenum. It's also the shortest version. We see it here in the purplish color. The middle portion of the small intestine is referred to as the jejunum. And then finally, the most distal and longest portion, which directly connects with the large intestine, is called the ileum. Notice that this is spelled I-L-E-U-M, not I-L-I-U-M, because if you were to spell it that way, you would be referring to one of our hip bones. Big difference there. Where the ileum connects with the large intestine, approximately here, is once again going to be a sphincter muscle, which we'll call the ileocecal valve. And ileo for ileum, obviously, and cecal for cecum. We're not really seeing this illustrated very well on this picture because it's focusing on the small intestine. But the very first part of the large intestine is this kind of sac-like structure, which we call the cecum. The cecum, oops, there should be another C here. The cecum will then turn into the ascending part of the large intestine, the ascending colon. We're going to then see the transverse colon, the descending colon, etc., as we'll see later on. I've tried to repeat this many times, but the main function of the small intestine is to absorb our nutrients. We don't see much absorption at all in the stomach, and we see hardly any absorption in the large intestine either. So most absorption occurs in the small intestine. And when we talk about water, let's start with water. Notice that the small intestine absorbs almost all of the water. 90% of the water is absorbed in the small intestine. A small portion is absorbed in the large intestine. So what about all the other nutrients? Well, by the time the food arrives in the duodenum from the stomach has only begun to digest the proteins. It has not really touched on the carbohydrates. Our mouth, the saliva, has begun the process of digesting some of the complex carbohydrates. We have a little bit of lipase produced by the tongue and a little bit of lipase produced in the stomach, but really nothing substantial enough to where we really have begun to tackle the lipids or the fats. And we have not at all mentioned any enzyme or any kind of a substance that can attack our nucleic acids. So all of that work, all of that chemical digestion still needs to occur in the small intestine, and that can then ultimately lead to small enough products that can be absorbed into the capillaries found in the wall of the small intestine. So anytime something has to specialize in absorption to where food particles, tiny small food particles, have to be able to pass through the epithelial tissue to make it to the connective tissue where the capillaries are in the wall, for absorption to occur, that kind of a structure needs to create a huge surface area. And the intestine, the small intestine, does that. It has three major surface area improvement, or I should say it has three major adaptations that very much increase its surface area. I'm going to list them first, and then I'll show them to you on pictures here on the next slide. We have the Flicky circularis, which literally means circular folds. We have villi, and then we have microvilli. Of these three adaptations that I just listed, the plica circularis, or in plain old English, the circular folds are visible on the inside of the wall of the small intestine. You can think, for instance, of an ice cream scooper that scoops out the inside wall of the small intestine, and that essentially creates these spiral indentations on the inside of the small intestine, hereby increasing its surface area. Now, we need a microscope to see the next two levels, with the bigger one of the two, the villi, followed by the microvilli. So if we look at a microscope slide of the small intestine, we see that right here. Right here would be the lumen. This is where the food particles would be located. All of this dark tissue is your simple columnar epithelial tissue in the mucosa, with a little bit of muscle tissue in there to form the muscularis mucosa. We can't really see that here. We have the submucosa, and then finally we have the muscularis externa here, eventually followed by the most superficial layer, which is serosa or adventitia, depending on whether we're intraperitoneally or retroperitoneally. Now, each one of these finger-like projections, let me change color to blue here. So this, for instance, is a villus, as is this one. So each one of these is a villus, and plural is villi. So that also increases the surface area. So the villi, as you can see here, are within these circular folds. The villi, these are your circular folds here, and the villi are these smaller invaginations. Now, microvilli are also often described as finger-like projections, but at an extremely small scale. So if we grab or if we draw one columnar cell, then its microvilli would be these finger-like projections of its own cell membrane. So here's the nucleus. You could assume that the lumen for this particular columnar cell would be right there. So you can tell that we're increasing the surface area by folds upon folds upon folds, or invaginations upon invaginations upon invaginations. Let's come back for a moment to the villi. Once again, let me illustrate the villi. So here we see approximately 1, 2, 3, 4, 5, and then part of a sixth one perhaps. On the diagram, we have just a couple, 1, 2. Notice that in between the villi, we see a little bit of an invagination, kind of analogous to the pits that we saw in the stomach wall. But here in the small intestine, we'll refer to them as crypts or intestinal crypts. The other thing I really need for you to notice is that each one of these villi has within it lots of connective tissue, which is very vascularized. But we also see part of the lymphatic system in each one of these villi called the lacteals. And it's these lacteals that are going to pick up the digested fats and not the capillaries. Finally, notice right here the muscularis mucosa, which is responsible for creating these villi and literally throwing the mucosa into these finger-like projections. Down here, where we see duodenal glands labeled, we are already in the submucosa. Here we see a very nice electron micrograph of microvilli. These are the finger-like projections found in the cell membrane of one single epithelial cell. This is our single epithelial cell with its nucleus, its Golgi apparatus, its rough endoplasmic reticulum and mitochondria, and here we see our microvilli. They're difficult to see with a regular compound microscope. It's basically almost impossible with a compound microscope to distinguish between cilia and microvilli. You really need an electron microscope for this. And consequently, under a regular or a compound microscope, I should say, they almost look like little hairs, but they are not. And unfortunately, we therefore refer to all of these microvilli as the brush border of the small intestine. So the small intestine's mucosa is simple columnar epithelial tissue on top of the lamina propria, which is very vascularized. It also has those lacteals in it. Just like the stomach, we see that this epithelial tissue is replaced every few days. There are always stem cells left at the base of the intestinal crypts. When the stem cells divide, one of them is going to remain a stem cell, while the other one actually will turn into a columnar cell that's going to be able to actively secrete depending on whatever function is needed. These cells are, just like in the case of the stomach wall, connected by means of tight junctions, so it's very difficult for substances to sneak in between the cells. The microvilli are very specialized for absorption. We have goblet cells that produce mucus to help make the chyme flow better, to make it stick together a little bit. And so what kinds of cells do we find in the intestinal crypts? You'll at times see that these crypts still get their older name, and that is the crypts of Lieberkühn. In these crypts, we once again find cells that produce hormones, and so we can just call them the enteroendocrine cells. But we also see cells that belong to the immune system, the intraepithelial lymphocytes, and even Paneth cells that can help with defending the small intestine against various pathogens, because they secrete lysozymes and defensins, which are two proteins we've seen many times, whether it is in the respiratory system or even now in the digestive system, and they'll keep popping up. Finally, we will also come across structures that belong to malt called the Peyer's patches. I'll point those out to you. The intestinal crypts that I pointed out earlier that sit in between the villi, so if this is a villus, we're going to see a crypt like so, followed by another villus in a perfect world, of course. These crypts are just the openings, and they lead to the intestinal glands. So this would be an intestinal gland, this would be an intestinal gland, very similar to what we saw in the stomach. The number of these intestinal glands tends to drop as we get closer and closer to the large intestine. So we see many of them in the duodenum, and then we see a lot less in the ileum. They again produce an intestinal juice of about 1 to 2 liters a day, and of course this intestinal juice is important in dissolving and buffering and moisturizing the contents of the chyme. What's interesting is that these intestinal glands already begin to secrete during the cephalic phase of the stomach. We see this happening throughout the whole digestive tract. The fact that the cephalic phase can already start digestive activities far distal from our heads. We also see that these intestinal glands are going to start producing more in response to being stretched as well as being irritated. Now, the intestinal glands, or the intestinal juice, I should say, it's slightly alkaline, so slightly lower, a pH that's slightly, not lower, I'm sorry, higher than 7, but it's not so high that it's not isotonic with the blood plasma. So it's almost the same pH as the plasma of our blood. It is very rich in water and mucus with some lysozyme in it. But this intestinal juice does not really have much of any enzymes. And so now you ask yourself, I hope, well, I thought the small intestine had all these enzymes that worked on chemically digesting all of our biomolecules. Well, we have a lot of enzymes in the small intestine, but most of them are actually stuck in that brush border formed by the microvilli. So they're not actually going to be part of that intestinal juice. So earlier I showed you a slide of the typical a histology slide of a typical when we look at the small intestine in a cadaver, let's say, it's next to impossible to differentiate between the duodenum followed by the jejunum followed by the ileum. We really have to look at the small intestine's microscopic anatomy under the microscope, therefore, in order to really distinguish between these three regions, particularly between the region of the duodenum and the ileum. And what we should focus on is the submucosa, because there are some big giveaways in the submucosa. For instance, in the duodenum only do we find duodenal glands, which I prefer to call Brunner's glands. These are glands that produce a bicarbonate-rich secretion, which can be inhibited by the sympathetic nervous system, by the way, when we're under a lot of stress, and the sympathetic nervous system tends to predominate for a long time. And ultimately, this could lead to duodenal ulcers, because if these glands are inhibited, they cannot make that alkaline bicarbonate-rich mucus such that the acidic chyme that keeps arriving from the stomach into the duodenum does not get neutralized properly. So in the duodenum we find Brunner's glands. Now in the ileum we do not see Brunner's glands at all. Instead we see lots of Peyer's patches. So let's take a look at that. So here we see a slide of the duodenum. We see the villi here, not the most beautiful villi, but this is the mucosa with a little bit of the muscularis mucosa. And this is then followed by our submucosa, which is filled with all of these big glands you see right here. And those are the Brunner's glands that produce that alkaline secretion that is very rich in bicarbonate juice. And then we see our circular layer and longitudinal layer that forms the muscularis externa. The submucosa of the ileum looks very different. First off, what we're seeing here is probably a bunch of food or sloughed off cells. These are our villi, which also are not as distinct looking in the ileum. And now we see these rather elliptical shaped structures. I've lost a little bit of resolution when I enlarged the slide. And those are your Peyer's patches. Very different looking from those glands that we found in the duodenum. Let's wrap up our discussion of the histology of the wall of the small intestine. The muscularis externa, as I've explained before already, is made up of your typical circular smooth layer and longitudinal smooth muscle layer. We find that the serosa, the peritoneal serosa, covers a good portion of the small intestine, but what we find is that a good portion of the duodenum, on the other hand, sits retroperitoneally and is not going to be covered completely by the peritoneum. The small intestine is characterized by both types of muscle contractions, and that is the muscle contractions that lead to segmentation and the muscle contractions that lead to peristalsis. For the small intestine, segmentation is the most common movement. Remember what that means, segmentation. It's the mixing of the food, as if you're preparing a ball of dough to make bread with. Once again, we see that the contractile activities of the muscle cells are initiated by the pacemaker cells called the cells of Cajal, which we find throughout the whole muscularis externa layer of the digestive tract. And they're going to need neural and hormonal stimuli to reach a threshold voltage. The depolarization waves are collectively referred to as the basic contractile rhythms, and they change a little bit depending on where we are. So as we start in the duodenum, we see that the rate at which depolarization occurs in the wall of the duodenum is about 14 times per minute, but as we get closer and closer to the large intestine, we're seeing that it begins to slow down to maybe 8 or 9 times per minute. So these rhythms are pretty set. The rate of depolarization can really intensify, but the actual rhythms cannot really change. And so this ultimately, even though this is segmentation that we're talking about, ultimately the food will slowly but surely make it to the ileocecal valve. What really gets the food moving in the small intestine is peristalsis. Segmentation helps out a little bit, but as I've mentioned before, the main function of segmentation is the mixing of the food. Peristalsis really focuses on getting the food to move down the small intestine towards that large intestine. And this is going to really take over after most of the nutrients have been absorbed, which makes sense. There's no point in starting to move the food out of the small intestine if the small intestine is not done absorbing the nutrients in the chyme. These peristaltic movements in the small intestine get a unique name. We refer to it as the migrating mobility complex, which is controlled by a short reflex, so we do not see the involvement of the central nervous system. It's kind of similar to what we saw in the stomach in that each new wave starts more distally compared to the previous wave, which kind of makes sense. Otherwise, we wouldn't be able to push that food down the small intestine. With the help of these peristaltic movements or contractions, we'll also see that old cells that have that need to be sloughed off will make it into the large intestine. Any kinds of bacteria, meal remnants, these peristaltic contractile forces will ensure that all of this stuff will literally make it into the large intestine. And it really is an important function to prevent any kind of overgrowth of bacteria. This takes about 2 hours, and notice that we're now mentioning another hormone called motilin, which is produced by the duodenum and ensures that this whole process of peristalsis occurs. So we just mentioned the short reflexes involved in peristalsis, better referred to as the migrating mobility complex in the small intestine. We even have a hormone that is released in response to neural stimuli. The hormone is called motilin, which can also contribute to the peristalsis in the small intestine. But we also have a long reflex that occurs, and we call that the gastroileal reflex. And this is how it works. When the stomach starts to become more and more active, we're going to already see that this impacts the ileum. So we're quite a ways away from the stomach, and we already see some mobility occurring in the ileum. Once there is some mobility occurring in the ileum, it's going to relax that valve, that sphincter that interconnects the ileum with the large intestine, and consequently food can enter the beginning of the large intestine called the cecum. So this is a long reflex. So this is going to involve sensory information to be sent to the central nervous system, and then from the central nervous system we're going to have to see parasympathetic fibers making it all the way down to the digestive structures. So this is called the gastroileal reflex. We also see that another hormone plays an important role, and that is gastrin. This time, the gastrin released by the stomach itself can again influence the activity of the ileum, which ultimately promotes the entry of food into the cecum because the ileocecal valve relaxes. So the ultimate result of gastrin versus the gastroileal reflex is the same, meaning that we're relaxing that ileocecal valve. So speaking of this ileocecal valve, once again, only so much chyme is going to be able to pass from the ileum into the large intestine. Kind of something similar we saw in between the stomach and the duodenum. Eventually that pyloric sphincter is going to have to close. We're seeing different reasons for why the ileocecal valve needs to close here, but essentially as the chyme starts to enter into the large intestine, it's going to build up pressure that works backwards onto or against the ileocecal valve such that it's going to force the valve to close. And so, since food has begun to enter the large intestine, I guess we're ready to move on to the large intestine. So that will be our next video.